Hello, and welcome to this Nature.com custom webcast titled Multicolour Digital PCR for ctDNA Detection in Breast Cancer. My name is Joshan Karpan, and I will be your moderator. Today's webcast is sponsored by Stilla Technologies. We'll begin the webcast with a presentation from Dr. Isaac Garcia-Morales of the Institute of Cancer Research London, and then end with a Q&A session. To ask a question, just type it in where it says type your question here and then press submit at any point during the webcast and we will answer them today. And now over to Isaac.
Hello everybody, thanks for joining on this webinar today. My name is Isaac. I'm a senior scientist at the Institute of Cancer Research in London, and today we're going to talk about ctDNA analysis in breast cancer. We're going to focus mostly on pre-analytical and analytical considerations during the talk. So let's begin. Breast and prostate cancer are the two most commonly diagnosed types of solid tumors. In the UK alone, over 55,000 new cases are diagnosed every year. Out of those, around 11,000 go on to perish from the disease. Around 27% of those cases are preventable. Excess body weight, alcohol, and physical inactivity account for roughly 20% of the cases that could be prevented with little lifestyle changes. In recent years, there's been major progress in breast cancer management. There's been an increase in the survival rate of those women diagnosed with the disease. At its stands is around 78%. The trend in improvement has been around 38%, which means that breast cancer patients survive double that they used to survive 40 years ago. The trend over time regarding mortality has seen a marked decrease. This stands around 35%. Still, there's around 22 to 25% of women diagnosed with the disease that go on to perish. It's this group of women which we are interested in finding solutions for the disease. One way to approach this problem is to molecularly and genomically analyze breast cancer patients. Efforts like the TCGA effort has shown us that regardless of molecular subtype, the most commonly occurring mutations in breast cancer are in PIK3CA and TP53. When it comes to copy number, genes that are commonly gained include PIK3CA and HER2, while genes that are most commonly lost include MAP2K4 and TP53. Historically, the way we have genomically and molecularly analyzed our patients is by using nucleic acids extracted from tissue biopsies. Tissue biopsies are invasive procedures, they are expensive, and the processing takes time. The DNA that we obtain from tissue biopsies is sometimes chemically damaged due to the procedures used for fixation and embedding of the tissue. Tissue biopsies also might not offer a whole picture of the disease, as we tend to take just one biopsy. In recent years, there's been a trend towards finding a solution for this problem. We have been developing new tools to analyze so-called liquid biopsies. Liquid biopsies offer a non-invasive assessment. They are less expensive than classical biopsies, with a much faster purification. We think they offer a whole picture of the disease that is not affected by tumor heterogeneity, and they are surrogates when anatomic biopsies are not feasible. When we think about liquid biopsies, we tend to think about blood. It is true that blood is the most commonly used biofluid. Other biofluids, like cerebrospinal fluid, saliva, urine, pleural effusions, and lavages, offer a different avenue for the extraction of nucleic acids. The most commonly investigated markers obtained from liquid biopsies are CTCs, or circulating tumor cells, ctDNA or circulating tumor DNA, and exosomes with their cargo of protein and RNA. Biofluids contain all the different metabolites that could potentially be used to develop biomarkers. For today, we will mostly concentrate on ctDNA. So, what is ctDNA? Circulating tumor DNA, or ctDNA, is fragmented DNA set by tumor cells undergoing apoptotic or necrotic events. This highly fragmented DNA is just the minority of the DNA found in circulation. Healthy and inflamed tissue also releases circulating DNA. This circulating free DNA released by non-tumor cells by the same process, i.e. apoptosis and necrosis, constitutes most of the DNA in circulation. When it comes to analyzing ctDNA, the number of events that can be investigated is quite vast. Mutations, copy number variations, methylations, all of these can be investigated from the DNA set by tumors into the systemic circulation. The amount of ctDNA that is found is pretty low, accounting for around 0.1% on the early stages of the disease and going as high as to 80 to 85% in later stages of the disease. In recent years, there's been a major push to develop ctDNA as a clinical biomarker. The clinical use of ctDNA is quite varied, and it could potentially be used at all the different stages of the disease. It could be used on screening programs for cancer detection, to diagnose the disease earlier, could be used to molecularly profile patients or to detect minimal residual disease following neoadjuvant therapy. It could be used to monitor response to targeted treatments, and it could also potentially be used to monitor clonal evolution of the disease. In order to use ctDNA in the clinic, we still have to overcome several major challenges. In my mind, the challenges faced to move ctDNA into the clinic can be broadly divided into three big groups: technical challenges, clinical challenges, and socio-economical challenges. When we talk about technical challenges, we are talking about how reliable is ctDNA for determining the presence or absence of a given genomic aberration. The clinical challenges center around the clinical benefits from those test results obtained from ctDNA as compared to those test results obtained from tissue. When I talk about economical and ethical challenges, socio-economical challenges, I am referring to as who gets access to liquid biopsy testing in the framework of intervention clinical trials. All these challenges are important challenges we are faced with when we want to use ctDNA in the clinic. For today's talk, we're going to focus only on the technical challenges we are presented with when we want to find a clinical utility to ctDNA. These technical challenges can be broadly divided into pre-analytical challenges, which center around sample collection, sample processing, and sample type, and analytical challenges, which mostly center around choosing the right assay to investigate ctDNA. So let's start with the pre-analytical considerations we need to take into account when we want to analyze ctDNA on a clinical context. We're going to talk about blood collection tubes and how to process them, if we should use serum or plasma, and which is the right extraction method to obtain ctDNA for clinical samples. When it comes to blood collection tubes, we have two different avenues for exploration. We have immediate processing using EDTA tubes, which stands as the gold standard, and we have delayed processing using tubes containing preservatives. In our lab, we have conducted a small proof of principle experiment in which we compare ctDNA obtained from immediately processed tubes, EDTA tubes processed 2 hours following venipuncture, against delayed processed tubes, which are extract tubes processed between 72 and 96 hours following venipuncture. The correlation in the amount of total cfDNA per mil of plasma obtained in this study was quite good. It's true that some of the delayed processed tubes had more DNA than their immediately processed counterparts. This is probably due to white blood cell lysis and release of genomic DNA. When it comes to mutation status interrogation, there was a very high agreement between those mutations detected in samples that had been processed immediately versus samples that had been processed with a delay. Those samples that were discordant were mostly samples in which the allele frequency of the aberration was quite low. When it came to mutation frequency and mutant copies per mil of plasma of those mutations investigated, there was a very high correlation. These 96 patients were patients from a metastatic set. So as we have seen, we can use immediate and delayed processing to obtain ctDNA from plasma. How we process our blood collection tubes for ctDNA work is of major importance. In the lab, we process EDTA tubes with a maximum delay of 2 hours following venipuncture, while preservative-containing blood collection tubes are processed with a maximum delay of 96 hours. We minimize the number of freeze-thaw cycles the plasma goes through, as well as the number of freeze-thaw cycles the extracted DNA goes through. There's currently a difference in the literature between groups using 1 or 2 spins. For 1 spin is the possibility of a reduced risk of contamination, especially when processing large number of samples. For 2 spins is the ability to remove more cellular debris, as well as potentially contaminating genomic DNA released from white blood cell lysis. This genomic DNA derived from white blood cell lysis that contaminates ctDNA is more obvious when we use serum. Processing serum requires a clotting process. During this time, white blood cells are lysed and they release their contents into circulation. Plasma, on the other hand, is a much more cleaner biofluid. Plasma is not affected as much as serum by white blood cell lysis it processes correctly. When it comes to the amount of circulating free DNA obtained from serum and plasma, we can see from these pictures that circulating DNA in serum is highly contaminated with high genomic molecular weight. This is a problem not presented in plasma. It is true that it will be advisable to use plasma to obtain ctDNA for clinical studies, but it's also true that retrospective trials have serum stored. Finding a way to obtain circulating tumor DNA from serum could potentially unlock all these samples. In recent years, the large number of samples available from clinical studies for ctDNA extraction have made extraction of this ctDNA using manual techniques a challenge. Recently, automated platforms have come into play. In the lab, in the metastatic breast cancer setting, we have conducted small experiments to compare automation versus manual extractions for ctDNA obtention. We use circulating nucleic acid kit from QIAGEN as the gold standard, and we compare it to 2 different automated platforms, the MagMax Kingfisher from Thermo and the DSP circulating DNA kit in the QIAsymphony from QIAGEN. As you can see, the correlation in the amount of total cfDNA obtained with manual extraction and automated extraction is quite similar. When it came to analysis of genomic aberrations in this small cohort of metastatic breast cancer patients, we look at the mutation frequency and the mutant copies per mil of plasma, comparing automation versus manual extractions. As you can see from the top graphs, mutation frequency using automated and manual techniques is pretty good. When it comes to mutant copies per mil, using automation and manual extractions, it remains very high as well. In our mind, automation can be used, at least in the metastatic setting, with as much success as manual extractions for the obtention of ctDNA. Let's move on now onto analytical considerations for implementation of ctDNA in the clinic. We're going to center mostly about choosing the right assay with the correct sensitivity, and we're going to talk about error-corrected NGS for ctDNA analysis, as well as digital PCR technologies that are currently available for ctDNA analysis. Choosing the right assay to achieve the desired sensitivity is of paramount importance. In the metastatic setting, where the required sensitivity stands at about 5 or above 5%, the analytical methods that we can employ for ctDNA interrogation are quite vast and potentially could include whole genome and whole exome sequencing. The applications of ctDNA analysis of this setting center around tumor burden monitoring and investigations of response to treatment. In primary disease, where the required sensitivity drops to around 1 in 10,000, the analytical methods that we could use become slightly more restricted. Now, we potentially could use digital PCR and error-corrected NGS. The applications of ctDNA analysis on this setting center mostly about response to treatment and detections of resistant mutations. When it comes to minimal residual disease detection, in which the required sensitivity goes as low as 1 in 100,000, the analytical methods are quite similar to the ones we use in primary disease, i.e. digital PCR and error-corrected NGS. Potential applications of ctDNA analysis on this setting are the detection of early recurrence. So why is it so important to choose the right assay with the right sensitivity? When we look at the total amount of circulating free DNA per mil of plasma in healthy or patients with one of the major solid tumors, we can see that the amount obtained remains relatively constant. It's also true when we look at different stages of the disease. When we look at ctDNA, the story changed completely. Now, ctDNA is not detected on healthy individuals, while there is a marked difference depending on the type of solid tumors, with colorectal patients having the larger amount of ctDNA in circulation as compared to ovarian, lung, and breast patients. The difference is now very marked when it comes to a stage of the disease, with early stages having as little as 1% of ctDNA in circulation. What is becoming apparent is that the current assays do not offer the correct sensitivity for ctDNA detection, especially in early stages of the disease. The frequency of cases with detectable ctDNA in early stages is as low as 40%. Different studies have reported different figures, with a maximum of around 70%. In the latest data, it is possible to see that the trend remains very similar. Very low detection in early stage patients as compared to later stage patients, and relatively poor in some of the tumor types, especially breast cancer. Developing new assays with the correct sensitivity is one of the biggest technical challenges we are faced with when it comes to inclusion of ctDNA in clinical trials. So how do we analyze circulating tumor DNA from liquid biopsies? We use the same tools that we use to analyze DNA obtained from tumor tissue. We use massive parallel sequencing and we use digital genomic technologies. Massive parallel sequencing, and in particular whole genome sequencing and whole exome sequencing, offer a higher level of complexity at a higher price. These techniques can be used for discovery. We don't need to interrogate a preconceived or pre-designed set of genes. When it comes to targeted sequencing, we decrease in complexity, but we increase in sensitivity. Now, targeted sequencing cannot be used as a discovery tool to identify new aberrations. We are using targeted panels to a set of known genes or known aberrations. When it comes to digital genomic technologies, and in particular digital PCR, there is a major decrease in complexity, but there is a major increase in sensitivity. Digital PCR cannot be used as a discovery tool. As for targeted sequencing, we are interrogating ctDNA for a known set of aberration. The use of digital genomic technologies have become more popular in the latest years due to a faster processing time at a reduced price. The two main flavors of NGS on ctDNA remain the same two that we use for tumor DNA: targeted capture and amplicon-based panels. Both of them have pros and both of them have cons, and depending on the amount of ctDNA that we have for analysis, we will be tempted to use one or the other. The pros of targeted capture are larger panels and lower PCR-associated error rates. The cons is that it's highly inefficient, it takes a long time to perform, and is relatively expensive. On the other hand, amplicon-based sequencing offers a relatively fast and relatively expensive technique with an increased limit of detection. But we can only use smaller panels that have a higher PCR-associated error rate and suffer from errors due to DNA composition. The major problem with these two techniques is the limit of detection, which is quite inefficient for identification of genomic aberrations on ctDNA, particularly mutations. In the last few years, there's been a major drive to the developing of error-corrected NGS for ctDNA analysis. As an example, using Avenius technology, which is a capture-based approach, we can see that now DNA can be barcoded to identify mutations from PCR errors. Using this approach, it is possible to call mutations as low as 0.1% using around 20 nanograms of ctDNA per assay. The use of error-corrected NGS allows for a much more deep analysis of ctDNA obtained from liquid biopsies. In the lab, we conducted a small proof-of-principle experiment using Avenius technology in metastatic breast cancer patients comparing matched tumor DNA to circulating tumor DNA. We picked more mutations on circulating tumor DNA than we did on tumor DNA. This probably reflects the fact that circulating tumor DNA is better for the detection of tumor heterogeneity. It is also true that some of the mutations that were picked on tumor DNA were not detected on ctDNA. This is probably due to sampling and stochastic factors. Having said that, error-corrected NGS for ctDNA analysis using targeted approach technologies remains quite a valid technique. When it comes to amplicon-based error-corrected NGS for ctDNA analysis, the approach is very similar to the capture one. The idea is to barcode your PCR products to distinguish true mutations from errors associated to PCR cycling. By using the Envision technology from Inivata and comparing it to digital PCR for a subset of clinically actionable genes, we found in the lab that the correlation was very high when it came to mutation detection. When it came to PIK3CA mutation detection, the agreement between Envision and digital PCR was very high. When it came to ESR1 mutation detection between error-corrected NGS and digital PCR, the agreement stood at 100%. This comes to show that error-corrected NGS, either amplicon-based or capture-based, are pretty powerful techniques that can be used for interrogation of ctDNA obtained from clinical samples. So, what about PCR approaches for the analysis of circulating tumor DNA? It's very clear that classical PCR is a very inefficient way of analyzing circulating tumor DNA and should not be used. The use of qPCR remains still a very popular way of analyzing ctDNA by many groups. The advent of digital PCR that could be thought as qPCR performed indefined partitions have advanced the pace of ctDNA analysis in recent years. The two main ways to analyze ctDNA by digital PCR are those based on physical partitions like the ones offered by Thermo or Fluidigm, in which ctDNA is partitioned into defined microwells and analyzed on those microwells, and emulsion-based digital PCR, in which ctDNA is encased into oil droplets, cycled and interrogated for the presence or the absence of a given mutations within those droplets. Companies like iNostics, Raindance, Biorad, and Stilla offer this flavor of digital PCR, which is becoming more and more popular. We have many ways to analyze ctDNA by digital PCR, either using emulsion-based or microwell-based techniques. We can use a single dye, in this case a DNA binding dye, to interrogate for two targets, a mutant target and a wild-type target, and by modifying the primers in one of the targets, being the mutant or the wild-type, we are able to separate populations based on the intensity of the signal from those two different targets. We could use two dyes to interrogate for two targets. In this case, the mutant allele is interrogated for using a FAM label probe, while the wild-type allele is interrogated for using a big label probe. In this way, we can separate populations of those mutant alleles from wild-type alleles and identify them in the plot. This remains probably the most popular way of interrogating ctDNA. Because the amount of ctDNA available for analysis remains very limited, we would like to be able to interrogate for multiple targets using a single assay. One way to do this is by using a single DNA binding dye to perform multiplex PCRs. By designing amplicons of varying length, is it possible to distinguish different populations in the plot and it is possible to interrogate for different targets within a single well. It is also possible to perform multiplex digital PCR using multiple dyes, in this case 2, by varying the concentration of the different dyes and the primers that compose that assay. It is possible to distinguish different targets based on the intensity on the 2 channels. By varying the concentration of the components of your assays, we have been able to develop multiplexes to the most commonly occurring mutations in clinically actionable genes. By using only 2 channels, we are able to interrogate for a maximum of 5 different mutations in a single well. Sometimes it's relatively difficult to distinguish these mutations, and it's only possible to call the presence or the absence of a given mutation. In order to deconvolute this, it is necessary to perform a second deconvoluting singleplex digital PCR. This, of course, requires double the amount of the material, and as I have already pointed out, the availability of ctDNA is very limited. Ideally, we would like to perform multiplex digital PCR using more than 2 channels. Recent advances on the digital PCR space have brought us equipment that goes beyond 2 channels. It is now possible to buy digital PCR technology with 3 channels. We can now interrogate ctDNA using 3 different colors, FAM, BIC, and CY5. In this way, it is possible to interrogate for more aberrations in a single well. Users of 2 colors digital PCR will have no problem getting used to 3 colors digital PCR. As you can see from this slide, the 1D plots are exactly the same, just with the addition of a 3rd channel. Again, the representation of the 2 plots on these 3 color digital PCR systems is very similar to what we are used to with 2 color digital PCR. In this case, we can interrogate in a single plot for FAM versus BIC, CY5 versus BIC, or FAM versus CY5, and identify these different mutations we are analyzing for. What these new systems bring into the table is the possibility to look at our plots in a multi-dimensional space. In this way, it is now possible to look at the plots on 3D and identify mutations on these 3 different channels. This allows for the identification of mutations in a much easier way when using multiplex digital PCR. One of the major interests in the lab is the identification of mechanisms of resistance to therapy, with particular interest to mutations on ESR1, which occur in around 20% of endocrine-resistant ER-positive breast cancer patients. These mutations cluster around a set of amino acids in the ligand binding domain and have been reported at a higher percentage in aromatase inhibitor-treated patients. These mutations occur exclusively on ER-positive patients. They center around the 536 to 538 amino acid on the ligand binding domain of ESR1, which encodes the ER receptor, are mostly monoclonal, and they offer a worse prognosis on those patients that carry them versus those patients that are ESR1 wild type. In order to identify these mutations, and due to the limited amount of starting material, we have developed in the lab 2 different ESR1 multiplexes. These 2 different ESR1 multiplexes interrogate the 5 most common ESR1 mutations. The problem we are presented with on these multiplexes is that it is very difficult sometimes to distinguish individual populations, requiring, as I pointed out before, a second digital PCR to discern which of the mutations is or are carried by that patient. Recent systems offer the possibility of performing compensation or tilt correction to try to identify this cluster slightly better, but we are still faced with the problem of multiple clusters occupying the same space on the plots. By developing these multiplexes using a third channel, the identification of these single plots becomes slightly easier. It is now possible to move some of these populations into the third channel, deconvoluting the second channel and helping in the identification of the single populations. Hopefully, this approach will be amenable to the interrogation of more than 4 or 5 mutations per well. But what we would like is not to stop at 3 colors. We would like to be able to use more colors, more channels, more detectors. We would like to interrogate for more mutations on a single well. This will not only help to bring down the price of the assay, but it will also help to conserve ctDNA. Ideally, what we would like to have is a set of equipment that is able to utilize as many colors as possible, and it seems that this is what we can expect in the near future, with different manufacturers bringing out different equipment with different capabilities. So let me finish by saying that, in my mind, liquid biopsies have advanced quite rapidly in the last few years, particularly the analysis of ctDNA and its integration as a biomarker. Several technical challenges still need to be overcome to realize its full potential. These challenges mostly gravitate towards sample preparation, standardization, and development of techniques with the required level of sensitivity. In my mind, multicolor digital PCR has the potential to position itself as a really powerful tool that could allow more complex analysis of circulating tumor DNA, especially when limited material is available. And finally, a word of thanks to all those patients that altruistically have participated on these studies: the Royal Marsden Hospital, the Institute of Cancer Research, the Molecular Oncology Lab, and the Ralph Lauren Center for Breast Cancer Research, as well as to all academic, non-academic, and industry collaborators and partners, as well as those organizations that make this research possible. I thank you for your time.
Thank you for your presentation, Isaac. It is now time for the Q&A. This is your opportunity to ask Isaac your questions. All you have to do is type it in where it says type your question here and then press submit. So our first question, Isaac, and this one asks, is there any liquid biopsy biofluid better than any other for a specific tumour?
Okay, that's a really good question, actually. I suppose that we used to take in blood, because blood is one of the easiest to obtain biofluids, mostly because we also carry other type of tests on this obtained blood. Depending on the location of your tumor and depending on the vascularization of your tumor, other biofluids might be better for ctDNA detection. For example, in the case of brain tumors, cerebrospinal fluid seems to be a better choice, mostly because we think that circulating tumor DNA does not cross the blood-brain barrier, so making it very difficult, if not impossible, to detect in systemic circulation. In the case of other tumors like oral tumors, saliva might be a better choice. In the case of tumors in the pleura, fusions might be better than blood. So I would say that although blood is the most, currently is the most used of the biofluid, and seems to be pan-tumor biofluid, depending on the localization and, as I said, the vascularization of your tumor, you might be better off using other type of biofluid.
Excellent. Thanks, Isaac. And our next question asks, does automation work as well in other settings as it does on the metastatic setting?
Well, that's a good question. It's a slightly tricky question. So we have tested automation in the early setting. We have also tested automation on the metastatic. And we have also tested automation on different fluids. It seems to work pretty well on the metastatic setting, at least on our hands, and offers comparable results to manual extractions. Now, we think it's not up there yet for the case of the early setting, in which very little amount of ctDNA is in circulation. Why it doesn't work as well on this setting when you have less circulating free DNA or less circulating tumor DNA, we are not clear yet. We think it's probably due to protocols being optimized by the vendors for a given amount of material in circulation, and we think they are not, they're not really there when that amount of material doesn't hit that mark. Now, for us, it's important that it works properly and it works in a standard way across all the settings. So we're working quite hard to actually see what is going on when there is no or there is little material in circulation, and why it doesn't work that well.
Great, thanks, Isaac. And our next question asks, is it always necessary to sequence circulating DNA, or can other techniques be used for tracking mutations?
Well, that's a quite nice question. I would probably say that no, it's not necessary to always sequence your circulating tumor DNA. It just depends what you're interested in identifying. If you are interested in identifying just a small set of mutations, for example, following a targeted therapy, you might be better off using a digital genomic technology like digital PCR, because it's faster and it's cheaper. Okay, it doesn't allow you to look for a lot of targets into a single assay, but what it does, it allows you to look for a specific targets with very high sensitivity. Now, if what you're trying to do is identify unknown aberrations, yeah, you definitely have to go down the route of sequencing. But I will say that this is going to be dictated by what type of study you are conducting and what are you trying to answer in that study. It's not always necessary to bring ctDNA sequencing on the table with all the cons, so the amount of material that it requires, the cost and the time it takes to do this. If you can do this with a much faster and cheaper technique, I mean, I think both of them have a role to play, and I mean, ideally, you should use both of them. Just it will depend what are you trying to answer, I think.
Great, thanks, Isaac. And our next question asks, can you multiplex multiple targets on 3-color dPCR approaches?
Well, the answer to that is yes.
I mean, the same way that you can multiplex multiple targets on 2 channels, you can do the same on 3 channels. Now, this all comes down to varying the concentration of your prime messenger probes in your master mix to move populations within those plots. And it also comes down to how well separated those plots are, and also those populations are within the plot. Now, the problem with this is, as any user of digital PCR knows, you start to get multiple populations with multiple targets on them. When you have only 2 colors and you have only 2 targets, you only get double populations. But as you start adding targets, you start getting populations that have 2, 3, 4 different targets. On 3 colors, you can picture the same scenario, but in a 3-dimensional plot, it'll be relatively easy to distinguish those populations that carry just 1 target. Now, how easy is going to be to spatially separate and visually separate those populations that contain multiple targets remains to be seen. But in principle, I will say that yes, it is possible, and actually that's what we all are after, is using multiple targets, using multiple colors.
Great, thanks Isaac. And just a quick reminder to our live audience, you still have time to ask a question. Remember, all you need to do is just type it in where it says type your question here and then press submit. So our next question from the audience asks, in your sort of opinion, which technique do you think is the most promising for detecting circulating tumour DNA?
In my opinion, I think, again, this depends. For discovery, I think what we would like to achieve, and this is going to come at a price, is error-corrected whole exome sequencing, okay? I don't think that's going to be easy to do. I think it's going to be relatively complex, and I think it's going to be relatively expensive to do. But I think that's going to give us a lot of information. Now, when it comes to looking for few aberrations, especially tracking patients following an administration of targeted therapies, I don't think anything is going to be digital PCR when it comes to sensitivity. I think in the future this is going to be an interplay between error-corrected NGS and the development of more sensitive and more powerful digital genomic technologies. That's my opinion.
Excellent, great, thanks Isaac. And our next question is talking about sensitivity, and it says if you're talking in terms of the number of molecules, what is the lower limit of mutant molecules that can be detected sort of using this technique?
So our lower limit is at 1 copy per mil. Now, that doesn't represent a single droplet, it represents more than one droplet. And, yeah, I think probably I think we don't report less than a copy per mil as we stand.
Sure, excellent. Thanks, Isaac. And our next question asks, do you need to use any special Taq polymerase for multicolored digital PCR?
No, you don't actually. One of the good things about at least of the system that's on the market at the moment, which is the NICA system from Stilla, is that it's open chemistry. Although the R&D department has optimized this for a given master mix, you are not tied to that master mix, which means you are not tied to any specific Taq polymerase. You could potentially get the system in your lab and optimize it to your liking using any master mix that you think will work better.
Great, thanks Isaac. And our next question asks, why do you think more colours would be an advantage, especially if analysis of the plots becomes too complex?
Well, it's going to become an advantage because it's going to allow you to look for more targets in a single assay. Okay. The problem that we have here is the amount of material that we have available for this kind of analysis. If you can only look at the target per assay, then it becomes very expensive in terms of material, how many targets you can analyze. The moment that you start multiplexing, it becomes slightly better because it means you can potentially look for more targets in one go. You could use just the same amount of starting material to look for 3, 4, 5 different targets. Now, as I pointed in one of the questions previously, the problem with multicolor digital PCR is going to be to actually analyze these plots that are produced. And I think it's going to center mostly about how we're going to discern those populations that have multiple targets in them.
Great. Thanks, Isaac. Well, I'm afraid that is all we have time for today. Before you leave us, we would really appreciate if you can spare a few seconds to fill in a very quick survey on your experience today. I would now like to thank Isaac Garcia Morales for his presentation and for answering your questions. And I would also like to thank the webcast sponsors, Stilla Technologies. And of course, you, the audience, for taking the time to be with us today. Remember, you can watch this webcast again at any time on demand at nature.com webcasts. Thanks for watching, and I hope you'll all join us again soon.